Okay, well, I guess we should get started. It's
Is it
11:02.
just me?
No, we've got
No,
Ankri's here, Kayfa's here. Let's see who else. We have
Doxy.
me, Big
Um,
Rabbit. Um,
yeah.
no more people RSVP'd, so they may kind of come in and out. But this is being recorded, so we will archive this on the Peer YouTube channel, so everyone can kind of see the recording. And I'll probably crop out Jihao's presentation so we can have a little shorter video for a scientific presentation. So, all right, we will we will get things started. So yes, welcome to Peer. The first data set that we're starting in Peer, which you'll hear about in a few minutes from Jihao, is this hippocampal formation subvolume CA3. So the hippocampus, I'm not going to go into the science right now because I wanted to just really briefly recognize all the effort that has gone into citizen science from Seung Lab over the years, and kind of appreciation of this next chapter that's launching. And I know you guys have been playing iWire for a long time. And I think we first mentioned Peter, what, in like probably 2017, 2018. It was called Neo in the beginning. We didn't have a data set for the longest time. And now the technology has come so far that there's like multiple data sets in the pipeline. So Peer is going to be more of sort of like a portal to new data sets that come out of Sung Lab. Um, so yes, let's just talk about iWire for 1 second before we kind of go into Peer. Um, I looked up this graphic from the day that iWire launched. We called it J Day, December 10th, 2012. And we had this ambitious goal of mapping a neuron in like, was it 1 week? The launch of iWire, it was like very
hmm
ambitious to have one neuron mapped in a whole week. Then I found this little time lapse of neuron growth, over 24 hours within iWire. And we were really excited to be able to map one neuron in 1 week at the time in 2012. Fast forward, I found this blog post from 2013. And this is in March of 2013, right? So 4 months later, after December, we were celebrating the sixth neuron that we had completed in iWire, which at the time was so great. We had a whole cell completed. We launched the sixth cell. Fast forward 5 years, and 250,000 people have joined iWire. We had these little characters, and the game had grown tremendously. And after 5 years of iWire, we had mapped 3,000 neurons or so. And then fast forward to today, it's been over 12 years of iWire. We have multiple data sets, from mouse and zebrafish, and together we've mapped over 6,000 neurons in iWire. But actually, iWire technology has sort of stayed the same. The game UI is fabulous and it's really fun to use, but the underlying sort of AI that assists the reconstruction hasn't really changed since iWire came out. But it has been changing at the lab and in our other projects. And I also want to say, if you guys have any questions at any time, feel free to just ask them with your mic or ask in chat. Okay, so you probably heard about Flywire. I should have put a screenshot of this Nature cover, but this is like one of, if not the top, like science journal in the world, Nature. And there was a whole special edition about the whole Fly Connectome that came out in October of last year. And there were a lot of citizen science contributions to this, 18,000 neurons. And 51,000 annotations. So, in iWire, you're like mapping cubes and the cells grow as you extend the branches. But in Flywire, you kind of have a whole brain and you can choose cells and reconstruct those whole cells, but you can also identify cells. So in Flywire, it's been a lot sort of looking at other resources and comparing with the literature, but over 50,000 cells were identified in the fly brain, and there's like 140,000 or so cells in the whole brain. Most of those were in the optic lobe, and Christoph Crook did a huge chunk of those. But this is a list of, these are all the flyers, all the iWirers who participated, who were actually co-authors. I'm getting a weird sound. Gonna mute. There we go. Yeah, so this, how many is this? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 people proofread 18,000 cells. And that's really just thanks to the improvement on the back end, on the AI side, and the automated segmentation. And now there's a new data set, the bank, which is the brain and nerve cord. So Flywire was like just this top part here, the brain, and then this is basically the entire central nervous system of the fly. And proofreading and annotation is active right now, and these same citizen scientists, some of them have been contributing to the bank. But this is all sort of hoping to be making possible this new project, PEER, which is so named for pyramidal neurons, which are the primary excitatory neuron of the brain. And PEER, we're kind of starting in the hippocampus, which Ziha will talk about. And we've made a lot of resources to assist with sort of the introduction to PEER because there is a different species. It's from a mouse, well, it's not a different species. Sorry, I've been in fly world for too long. It is a mouse, but it's a different region of the brain, so the cells look quite different from retina. And the controls are different because it's sort of a newer infrastructure platform called NeuralGlancer, and it's a new branch of that called Spelunker. So the controls are unfortunately different than iWire, and some better, some worse, but it definitely takes a little bit getting used to. So Marissa and Celia have spent a lot of time making resources to kind of help get folks oriented with it. But that's a brief intro and history of citizen science at Seung Lab. And now I'm going to pass it off to Ji Hao, who generated the Flywire dataset and has generated this new CA3 dataset for PEER. So thank you, Ji Hao. I'll pass it off to you, and I'll
Right.
stop sharing.
All right. Thanks for your introduction. Let me get up my my slide. Trying to find the right window. All right, you see my slide?
Yeah, but we're also seeing next slide and click to add notes.
Uh-oh, I don't have. I guess, let me try.
Isn't it the button down at the bottom, if I remember correctly?
I mean, I guess we'll just do this. Should
Yeah,
be fine.
That's fine.
All right. It's good to see everyone. I'm Ji Hao Zhang. I'm in my PhD, I kind of, worked for a few years on the microscopes and the data acquisition that eventually become Flywire. And I joined Sebastian's lab a few years ago and started to work on this brain region called hippocampus. So I think today what I'm going to do is actually I'm going to try to tell a couple of stories that I think really inspire hippocampus research and why we are focusing on this region. So hippocampus is a brain region in mouse and in humans brain that is known to be important for memory. And one of the most inspiring case is this patient called HM. And HM, later was revealed to be Henry Mollison, and this is a landmark case in neuroscience because in 1953, the patient HM, he has severe epilepsy, and then he underwent something called a temporal lobectomy, which is a procedure that involved removing part of the so-called temporal lobe, but involved most of the hippocampus was removed. After the surgery, his seizure was much better, but there's an unexpected and profound side effect, and it's called anterograde amnesia. Basically, he couldn't form new memory. And I don't, there's a lot of fascinating video of this patient. I can try to play one of that, and let's hope it works. If no, I can post
We can post it up later, yeah.
if no, I can post up later. Actually, I have it here. It might work. Let me share. I just need to share. Mhm. There you go. Yeah, so this is, this is called The Man Without a Memory. And I don't know if you can see the video sound.
No, I'm not hearing sound.
Not hearing the sound, but you can see the
Yeah.
video.
Yeah.
Well, I guess there's the script. So this is an interview with this patient. It's only a few seconds. Okay, I'll post this video in the chat now, and you can have a look later. We can post it later. There's a lot of fascinating video about this story. The other, so it's really inspiring that this is a region for really for memory. And the other story I would say is that um, in the I think this is in the 90s in London there is um the London cab driver has this famous rigorous test called the knowledge, right? Which basically require memorizing thousands and thousands of street landmarks and road within the city. And in the late 90s and early 20s, there's these studies that basically use MRI to scan the people who take the knowledge test. And it turns out that the people who do really well on the knowledge test and spend years studying has larger hippocampus. And the ones who fail has like smaller hippocampus, basically. So, this is sort of known as another another important functionality of hippocampus is spatial navigation, basically. Um, and in the and in terms of the science, from this is I guess this is work in the 70s, and later on in the 2000s, and then it was awarded Nobel Prize in 2014. And basically, people stick electrodes in the brain and recording from hippocampus cells. And what they found is that there are these cells that basically encode your encode space. It create a map of the room that you experience in your brain, basically. And these are called place cells. Um, and these cells are only active when you're in a specific location in the room. And therefore, you have a mental map of your surroundings. For example, if you're walking through your living room, certain place cell will activate to represent that specific location. And, so these are all sort of inspiring study. And the other thing I would say is that it's known that a lot of dementia, Alzheimer patients, one of the regions that's earliest to be affected is hippocampus. And some of the things that you would hear from people, that one of the earliest symptoms of Alzheimer patient people is that they forgot where their keys are, or forgot something that just happened um, a few moments ago. And so, this is a brain region that is very important for memory. It affects us um, in terms of the damage of it will basically affect our spatial navigation and memory. So these project is basically start with we wanted to start mapping the circuits of hippocampus. So the beginning of this is this region called CA3. This is a sub-region of hippocampus. I took a piece of tissue from a mouse hippocampus. So it's like here. And much like iWire and Flywire, scan it and image it and collect the data and reconstruct it. And our team reconstruct it in 3D. And now the data set is here. So the goal is to reconstruct circuit in hippocampus. Right. So that's my introduction for sort of the motivation behind the data set. I'm happy to answer any questions, details about, if you have any questions about details and other things, please, let us know.
Anyone, you have any questions, anyone? I've got one. So the grid cells, every time you're in a new environment, do they sort of reorient themselves to the, like if you walk from one building, to a coffee shop down the street or something, do the grid cells, like how do they reset somehow?
So it's known that the place cell would reset.
The place
So
cells.
you would have the place, but the grid cells, they would I would say they would change, but they would still similarly have a set of grid, and the grid will be at the same scale and orientation. So it's very interesting.
That is really interesting.
But the place cell would totally reset.
So what would happen if you just spent a whole day going into, I guess if you, I would just think if you just went from one place to a new place to a new place to a new place, just every time they reset, but somehow they recognize it, like because you were everywhere you've been, when you go back to, you remember that you've been there before.
Yeah. So what's interesting about this, and this also has to do with the patient HM, is that he has memory of things happened before the surgery, right? So the idea is that this has a map of the space that happened recently, and then when it becomes what we call long-term memory, it goes to somewhere else, cortex inside the brain, and stored there. So temporarily, for a few days, for some moments, you have memory of this room in the hippocampus of your brain.
Wow. Okay, I have one last question, which is how does, so say you have multiple rooms that are connected, right? Your brain is also not only mapping, like being aware of where you are in the room, but it's understanding where things are relative to each other, like being able to navigate from A to B, for example. Is that also place cells and grid cells? Like the relative position of one place to another place versus like your place within a given place.
Yeah, so I think this gets to a kind of a frontier of research in the field that has to do with the questions of, is it really for just specialized for space, or is it really for any kind of memory relationship, right? It could have and I don't think we had a clear answer. um but in general, if you have multiple rooms that are connected together, um they still have a similar place field firing, place field activity with each of the room and including the hallway between it. Now it would start to get tricky if you can construct something that is so-called non-Euclidean. It's that basically you have things between room, things of different room, like you described, things of different room. But the connection between them are sort of, relative. And, I think we don't have a very clear answer to that question yet. How specialized it is for space, when it become really a little bit non-space and more like your episode, what we call episodic memory, which is your memory of kind of everything, including space, but more than space.
What is the actual definition of episodic memory?
It's something that a person experience. So space is an important part of it, is the context of it. But include the context, the other context in which it happens, or sound, right? And then other components of it.
Like the activity of the organism within space. Okay, that's great. Thank you. These are really interesting questions to explore, and so different than what we've been asking, like of the retina, and also like of the questions from Fly. Okay, we have a question from, I'm sorry, probably not going to say your name correctly, but Yenadi Guskov. Do we expect to find anything specific in terms of the connections between the cells in the hippocampus, or do we not know what to expect yet? I think they're asking like what kind of things are we expecting to find in proofreading this data set?
So you probably know a little bit about all the amazing things that has happened in Flywire and Flywire. Now the key thing is that nobody had mapped, having tell this story about how important hippocampus is. Nobody has mapped hippocampus in the kind of detail and resolution with the kind of technique we use for iWire, Flywire yet. It would be our first time of mapping this really important brain region. So I think there's gonna be tremendous amount of discovery that we can find that could inspire model or how we think about how these memories are stored. I mean, I would say that the dream would have been that, is there something that this connection could encode the memory, right? That would be sort of one of the ultimate dream. And is there anything specific? I mean, connectivity between different type of cell and how it, how it could, um, encode the storage of memory. I think that's, yeah.
We've got some more. Hold on, some more chat. Are we
Anki
expecting?
was asking.
Yeah, Ankri
Yeah.
asked, are we expecting to find complete cells in the hippocampus, or will a lot of them also travel to other parts of the brain?
That's very good questions. Compare the size of a retina, a fly. Hippocampus is a brain region in a mouse brain that is really large. The beginning volume, which is what we are sharing with this effort, it's the cell would be kind of partial.
Here, you can see. There you go.
So these are a set of cell, you can see these large cylinder-like things are soma in the center with their dendrite on two ends, the basal dendrite on sort of the bottom and the apical dendrite on the top. However, so this is partial, but we have a lot of effort to rapidly increase the volume of hippocampus and hippocampus related region in the next it's really this year and in the next couple of years. I can tell you that there are already a larger volume that will be coming at some point in the hippocampus related region, hopefully later this year. And then the goal in the next few years is to map a large part of maybe almost a whole of hippocampus. Now traveling to other parts of the brain, whole mouse brain is where we also optimally wanted to go. But I think it's more like a, 8 to 10 years effort in the next, that's coming in the next, I would say 8 to 10 years.
I was just showing some of these. That's so exciting. So exciting. I lost my Zoom window here. Were there any other questions?
No, I think we're good. We'll be slowly granting access to peer for those of you who are already familiar with Flywire. We'll fast track you, and then we will have a method for getting folks onboarded who need to kind of learn the tools for Spelunker, this new software that's a little different from iWire, and get you access that way. So that's kind of a diversion. Um, as I think Amy you said, we're still early on with this, so part of it is that we will kind of be hopefully incorporating features and making things a little bit better on with this version of the software. That's pretty much at this point, I think the standard uh, software used in connectomics at this point for full data sets. So
So, and this one's not at the peer URL, but we did at least put user profiles in for peer. So you'll be able to see how many edits you made and how many cells you completed. And I saw one other question that is probably a good segue to Marissa here.
Mhm.
When I visited an early version of Flywire, it wasn't clear. I didn't understand what we were supposed to do, which is a fair question because it's just like, here's the brain. Go proofread it. So, unlike iWire, when we complete or fix something that the spawner didn't do right, what's new about the system peer? So, we're actually starting peer with two sets of neurons that need to be proofread. And one of those is launched in this. So we put a new blog up for peer that has some resources. It's just blog.peer.ai. So what Jihao was talking about, well, so there's a cell guide that we created that basically highlights some of the main neurons that you'll find in CA3. And these are the uh, thorny pyramidal neurons, so-called, because they have these little bobble type things on them. And those are uh, these crazy sort of spines that have these gigantic synapses with these axons called mossy fibers. And one of the things that we're launching Peer with is this Quest Challenge 1, which is to map the 400, that's not just mossy fibers, but the 400 presynaptic axons, so the neurons that go and form synapses, onto these pyramidal neurons from the dataset. Did I say that right, Jihao? I think I did. Um, Yes, so once you have access to Peer, we have it organized on a spreadsheet. I'm sorry, we're going back to the pre-Scouts log days, guys. We should open source a Scouts log for NeuroGlancer, really. But you'll be able to I don't know where we don't have the spreadsheet in here, but there's a spreadsheet to organize this. But maybe this is a good point for me to stop talking and show Marissa to pass off to Marissa with one last thing saying that we had a blog and it got we lost it, and so we rebuilt it. But so now the cell guide is missing some of the NeuroGlancer links, but I will add that back. So we'll have a NeuroGlancer link to. Oh, no, maybe it's not. Never mind. We added it back. So scratch that. They're all there. In the cell guide, there's a link where you can see like interactive versions of all the cells and structures that you'll find in Peer. So with that, I'll pass off to Marissa, who can really show you around.
Okay. All right. So where are we at time-wise? So
11:30.
yeah. Okay. All right. Let me start sharing my screen here. Okay, so most of you probably know me as Sork.m from iWire, and that's or M as I sign off my emails to you all. But I'm Marissa Sork, and so here's kind of just a basic overview of. I'm not going to really get too much into the weeds for some of the shortcuts for how to do things, but just kind of what the task overflow is. The first thing though is as Amy was we were talking about the anatomy for CA3. And I'm going to kind of just do a brief overview of what the whole data set looks like, just so when you get in there, you can see kind of what's going on. The first thing is you kind of notice from the pictures that everything's cut off. It's a very thin data set, but it's wide across. So here I've kind of just briefly zoomed out, and unlike iWire, right, where we only see a small little piece of the data set as a whole, we're seeing the actual whole data set here. The really cool thing that I think is pretty awesome that you can see here is if you can see this band of somas right here, this curve. And if we were to go over and look at where we are in the hippocampus for this little piece here, so here we have an image of the hippocampus. We're over here in CA3. This is the one that we're restarting in. And you see this curve here in the hippocampus. If we imagine we'd turn this image a little bit 90 degrees, that's the same curve that we're actually seeing over here. So it's kind of really cool to be able to really, really distinctly see that and identify that. And these are all the pyramidal cells that have been already reconstructed. There's about 200 of them approximately. And so now we're going to be, the goal is to get kind of the counts and see how many, synapses are coming in and everything like that. So we're looking at the presynaptic partners. So all those axons that are coming in from outside the data set and making connections with these pyramidal cells in the middle. And you can kind of see that here when we look at this basic just sketch of connections in here. So you can see there's like these very long fibers here. These are mossy fibers that are explained in our cell-guided anatomy. And then also you can see where there's other various cells probably poking in with their axons. And also, this is why Jihao talks about we'll start at different, this particular region within the hippocampus. They're broken up into these kind of four main regions. So just another little overview from where we're starting at and what we're kind of interested in doing and reconstructing and what we need your help with proofreading. So we have a spreadsheet here with some helpful links at the beginning, and we'll share this along. We also have a little mini leaderboard for how many you get done. And here we have those 400 segments already pre-put in because we're able to run a query. Jihao and his team have done that to find all those presynaptic partners. And so now we need to clean them up. So I'm going to just go ahead and copy this segment ID here. So you can kind of think of it as a cube ID if you're not, as familiar with this. Similar to how you have a cube ID, this is kind of the way that we have it where it's attached now to the individual segments or within the data set as a whole. Because you can think of it as a big scaling up from what we're used to looking at in EyeWire. I'm going to go over here into my NeuroGlancer link here. And I'm going to paste that in. So here is kind of a default view. We have our EM here on the left, and we have our 3D view over here on our right. I'm going to paste this in. I'm currently in a tab here, and we'll talk a little bit. As I said, I'm not going to really go into some of the more like navigational things a little bit here. I'm going to kind of just overview what the task flow is like, and then obviously during training and things like that. And I'm going to be doing a call, a Zoom next week, and I'll definitely get a little bit more into the weeds then. So I'm going to paste this segment ID over here into this list. You can see it's now green highlighted here. If I want to visualize that in my 3D and in the EM, I'm just going to click this little star button here, or the I button here. And that means I've selected it and added it in. So one thing that'll happen is you'll paste it in and you'll maybe not be in the right part of the data set. But the nice thing is you can just zoom out and there's our little segment here. So this probably will look more familiar to you, right? You're used to branches from what you might be seeing in EyeWire or, in Flywire too. For those of you who already been in Flywire, this probably, you're well familiar with this. So I'm going to actually be proofreading everything, including, it's pretty obvious, right? I think most of us know this is a merger, right? We see the kind of the crossing here, perpendicular. Pretty sure that's going to be a merger. However, for these segments, we don't have kind of what starting point we want to start at. There's no like, hey, we want to proofread only this big branch, or hey, we only want to proofread this little branch, because we aren't starting from like a soma and then growing it out like we do on EyeWire. We're starting from kind of like, hey, this segment we know is synapsing onto a cell that we're interested in, and we need to reconstruct it. So what the goal is we'll reconstruct both these segments that we see, even though we have this merger. So we're going to reconstruct everything we initially see on here. I'm going to go in here and I'm just going to kind of check off that I've, finished it. So sometimes they're very, they've already hit the edges of the data set. Sometimes they need a little bit of extra help. In the case of this one, it looks like for our big segment, we might not need to do too much work. One thing I'm going to do is you can create how do you keep track of where you've looked and things like that? Well, we have things called annotations here. So here I've just right-clicked on my plus sign. I've got a little annotation tab here, and I'll just rename it like, dataset edge. And that'll kind of let me know, hey, I've completed and looked at this branch. So what I'll do is I'll just change this color to red, just so I know it's like a stop. So I'm just gonna right click to just jump myself right to the end of this branch. No, I could panel on it if I need to really look in at it. But in this case, this is pretty smooth as we look at it. It's pretty good. And the AI has done a good job. It's come leaps and bounds since iWire's AI. And in terms of segmentation, you can see it's doing whole things. So here we have a blank slide and then a continuation. So this is a common error within the data set and go from there. So, oh, it's about 2,000 somas. Oh, I'm seeing chat over there while I'm like talking. Anyways, so basically, I'm gonna try continuing this. So I have this one black slide, and we've seen these things in iWire. It's usually where we see like a little striation, a little bit of a misalignment or something. And this is it's a jump. So for this one, the nice thing is if I zoom in, you can see there's a little like we're seeing all the intracellular organelles and mitochondria inside the cells, which is a lot different from iWire. I know that might be a big jump for folks, but it actually helps a lot with kind of seeing things and noticing things. So for this one here, it's kind of pretty obvious. We have like this very big structure down here, and then a little friend in between, and then our cell. So we can kind of just I can almost even just hover over it and it's, doing that. And I can look and I see like the same kind of little intercellular structure right here. So this will do that. And I'll just double click, select that little segment piece, and oh, look at that, our branch is, grown. We can look here too, and see that in the 3D, the diameter, so the thickness of the branch, it's, matching the flow is still going correctly. So, when you're flagging things as a scout or fixing things as a scythe, you're familiar with, noticing when a branch might be kind of looking a little weird, like it's growing the wrong direction or something like that. And that's the same thing that we see here. It just applies the same, it's just at a little bit of a bigger scale. So here we have that branch all the way grown up, and it looks nice and smooth. It's the same kind of structure and thickness. We're seeing like these, not necessarily kind of boutons, but they're like blebs, as we kind of can call them. And they are boutons in a way. And what I'm going to do is I'm going to just jump right on up to the end here, and we're going to scroll. And oh, it looks like we're getting a little bit at the end. It's a little messy. And I know I'm getting near the end here because if I look here, if I zoom out, you can kind of see where we have the top of the data set here. So we have the volume here, like these little data set bounding boxes. And also, if I keep scrolling just a little bit more up, now I don't have any more data. It kind of ends. So kind of once we get to this edge here, it's around like z equals 100. One thing I kind of know is, and I'm going to post this into the proofreading guide too to kind of just help you, is I can look at my numbers up here for the coordinates within the data set, and I see that it's saying like z equals 100. And we're familiar with that from iWire. If you were Scythe, I think we've talked a little bit about like when we reach that one particular area near the edge of the data set, I think it's z equals 420 or something like that. And I would always be like, let's not proofread anymore. It's too much of a jump. We can't really do anything about it. So that's kind of one way to do it. I could add in maybe see if I can get a little bit more, but you can see after I get off past this area, there's suddenly like we have like this big, pieces of things happening over here. We have a soma, so it's changed kind of, there's too much of a dramatic shift in the data. So I'm going to use my annotation point over here. I'm going to click this little dot next to my red and say, hey, I have found the dataset edge here. So I place a little point and now I know I've checked that. And now if I zoom out, you can see, hey, I've tracked that. I'm going to go down to the other end over here. Let's just kind of finish this whole long branch part out before we go to the fork and remove the merger. And let's see if we have a continuation down here. Well, No. Yeah, we don't really necessarily, have a flat top in the 3D view. Um, I mean, it's kind of just how it goes. So you can turn on your slices or turn on your slices here, please. Sections. No, I'm in black, that's why. And you can see here, your slices do kind of angle here when we kind of go through the data set here. And you can see now I'm getting just nothing but black slices. So this is the edge of the data set. Um, I'm going to go ahead and put a data set edge here point, and I'll just drop one right there. I highly, if you need a little bit extra help for where you are and everything, turn on those sections, look at those slides that are being visible in the 3D. You can obviously, that, is something we do in iWire 2 as well. You can toggle that on and off, right? Personally for me, especially because I'm trying to just kind of do this quickly, I'm turning them off just so you can kind of see the structure a little bit more in the 3D. Okay, so we have these two areas here. I'm gonna go ahead and add these two segments real quick here. So I'm gonna merge them together. So that's a little bit of what is a little different from iWire, right? Is that previously you would just add the segments and then submit the cube and they would be connected. Well, here, because we have the whole data set for ourselves and we're, growing everything, we actually will just actively merge it together. So I'm going to hit M for merge, and you'll notice down in the bottom left of my screen here, it tells me I'm in merge mode here. It says merge segments, and I'm going to go ahead. I'm going to use Control left click to place a, oh, I need to get out of my annotation tab here. That's something to be aware of. So I want to make sure I go over into my segmentation tab here. So in this case, I'll explain the tabs here. So this is your EM slices. This is your kind of segmentation tab. It's what gives you the 3D and the coloring within your EM. And then here, this is just another thing telling you where all the nucleuses are, but we'll ignore that. And then this is that annotation tab I had where I created for my notes. I'm going to select this, and oh, here we go. We have our segments back here, and you can see my yellow segment and my green segment right here. So let's merge these together. We're going to use Control left click, and you can see now I have a little line to draw between the two of them. And I can do this in the 3D or the 2D, but I'm just doing it in 3D. No need to really do much, I can just do this. You'll see here I have that yellow. It's being changed to the green, and I'm going to hit submit. And it's gone through, it's done that. It's joined them together. You have a kind of a new segment ID number and a new little color here. And it'll render it all back in. It takes a couple minutes sometimes to do that. And there we have our branch. It's all joined together here. So let's go over here and we'll look at our merger. So here we have a fork, but if I look close here, we can see, it looks really good in the 3D. It's very smooth and natural. And once again, we're looking at the overall structure, right? We're looking at these branches. And you're just going to scroll. And okay, look in the 2D here, you can just see it naturally splits apart. And, I can right click and that's centering on the branch I already proofread. I'm going to go over here onto this one. So we'll follow this out. It looks great. We're seeing, it's very smooth. It's having the same structure pattern here. We're going to go definitely to this part here. Okay, I know this is a merger, right? We can see it. It's a very tiny little merger. Look at this. It's just a little tiny point. Like, oh, accidentally got merged together here. So what we're going to do is we're actually going to cut this off. So we can split things apart. So previously, right, we would have to kind of, deselect the segments within iWire and be like, no, these don't belong. Well, here we're going to just say, hey, these need to be cut apart. So I'm going to use C for cut. And you'll notice that, hold on, let me get out of merge segments. So to exit merge, I'm going to hit M. I'm no longer the merge segment, no. I'm going to hit C to cut here. And you'll notice that it now is saying, I think it says multi-cut segments. My my Zoom little overlay is on it. And you'll notice that I have like something called an active group here. And that's how I'm going to tell the computer this belongs to one thing and this belongs to another thing. I'm going to place points once again using that control left click. 1, 2, 3. Say, hey, this is all the red branch here. And I'm doing this in the 3D. I can also do it in the 2D. But for this, it's pretty obvious. They're two separate branches here. So we can just place them in the 3D. I'm going to swap. So I'm going to swap over to the blue group now. So that's going to be my other branch that I want to say, hey, this is separate from this other red area here. So I'm going to put some blue points in this branch. And then what I can do is I can just hit submit. And ta-da, they're split apart. And we can see that too. So I had my cursor right where the kind of little brief little, like almost like little kiss that was happening between those two that was causing that merger. And you can see it in the 2D here. It's now split apart. We have two different colors and our branches are good here. So what I'm going to do though, because this was part of our starting segment, and at the beginning, remember I mentioned we're going to proofread everything we kind of get started with. I'm going to just hit the I button in invisible this segment for a minute, just so I can focus on our original, our big branch here that we already proofread parts of the ends. I'm going to just go to the end here and we'll see real quick. Can we find a continuation? And it looks a little fuzzy here. So you can kind of see like we're right here at the end. It's getting a little fuzzy. And that's partially because we have a dark um, like organelle here or mitochondria or something or other, or another branch here. But I'm going to use nearby. And here we have like kind of this friend like that's right on top here. And we can see our little blue piece kind of getting squashed underneath here. So you can you have to scroll through your 2D once again to look for that video. So just similar to iWire, right? We're going to kind of use things that are around it to try to find where that jump is. And here, okay, that looks good. See how it kind of matches style-wise. So let's do a little more. And okay. So there's this piece here that's kind of disappearing into, but I can't, do you see how when I'm hovering, it's not giving any segmentation here? Sometimes it's missing segmentation in the EM, and we kind of have to just wait till we can hover and click something next. In the case for this one thing is that's really handy, as I mentioned, is you can use those interstellar structures. And um, so here, you can see there's a little line. And this line continues. Do you see that line? Just continuing along. And that tells me, okay, this is going this way. And it's nice. It gives you kind of that little help. This is actually a microtubule, is what it's called. That's inside the cell, if I remember correctly. And that continues. So I'm just going to click here. And oh, hey, that looks nice. It has that nice line still continuing inside the cell there, or inside the EM and the branch here. And also look at the thickness. Thickness looks good, right? We have still that kind of, it's thicker right here, and it's flowing correctly. Like it's taking a turn here, but it's still very similar. I'm gonna just kind of jump up here, see if we can find a little bit more. Oh, it's going this way. Another slide here. Okay, we're taking a dive. And ooh, this is a little jump here. And okay, let's see. I think that's the wrong piece. Ah, all right. This is a little bit where we're going to get messy. And so you can see here, this is where we have things that like we get these knots where the computers really just put these together. And that's where we come in. This is kind of like, hey, this is where we as humans need to come in and untangle this. And it's only high level. There's nothing like inside the branches that you're going to be really adding, is a good question that you asked, Gigi. Um, you notice there's not really much for me to add inside of these. And you'll notice that I couldn't select certain things, and I'm not adding in these like missing holes here. It's not really relevant. Like, it's totally surrounded in this piece here. So we don't need to even worry about adding those in. We want just kind of the long structure here. So for this one, I have a feeling I have a couple mergers in here that I'm going to have to take care of. So let's see here. One, if you get too many similar colors, see we have two oranges that are very similar. You can go over here and change that. Let's make that yellow. There we go. And my cat is acting up in the background right as I'm talking through this.
Your screen right now.
Hey, he's mad at me. All right. So sometimes what happens when I'm doing these demos is I get in a sticky spot and I'm like, oh, this isn't very obvious to me. And everyone else like watching is like, oh, hey, we got this. So
Like
let me,
I'm not seeing your screen right now. Are you guys seeing her screen?
are you not seeing my screen?
No, I
I'm
was
seeing it.
you see it? What the heck?
Uh-oh.
Hey, you guys, G House said he can see it. Okay, well, all right, never mind.
Yeah,
I'm glad
Cafe
Right,
is saying that too.
I was over there.
So
Oh, vlog. Okay, sorry.
yeah, you're good. You're good. Okay, so I think we have, this is going to be a tricky situation here, and we're going to have to kind of scroll through and see if we can find that right continuation. However, what I'm going to do is just for brevity's sake, so we don't go too long, we'll just be like, we'll figure that out in a bit. So I'm just going to kind of hide things a little bit here. So I'm just removing those segments right now. I'm going to quickly just merge these good pieces together because I know they belong. And you can see here, I can merge multiple segments at once. So you can merge up to 10 segments all at once, which is really nice. You don't have to do them individually. And I just kind of boop, boop, string them along. I'm in that merge mode. I'm hitting enter. You can see it's trying and done, and it's merging those all together for you. Um, all right, and so that's done. I'm gonna exit my merge mode by hitting M on my keyboard. And just so we know, well, we can also proofread this. Celia has a longer video that we're going to post up of really truly proofreading like all these branches here. So what I would do is I would come back and also proofread this branch. And here, once again, we have that thin connection going through. We really have to kind of look for where there might be a continuation. And it's kind of tricky. So we have this blue piece here where this piece is getting stuck, but it's probably actually this piece right here. And that makes sense. So here we have a jump, and you can see here it has a little fuzziness where it gets into this kind of black overdyed area here. And then it kind of goes down. The other thing we can check is we don't see anywhere else for this orange to go, and it continues nicely from this. So that's a kind of a way that we can check for the continuations. And so then we would be growing this piece out as well. Let's say we're done. Let's say we're all done. I'm just so we end on time here today. We have our main segment that we started with here, and it's hit the edge of the data set for everything. We'll pretend we have. What we're going to do, how do we submit it? Well, it's already live in the data set. Every edit you make is going, it's actively updating. You're editing the actual live data set when you're in here. So multiple people might be editing certain things all around you. That's very possible. So you can share this though, and we'll save the link so then we know it's done. I'm gonna hit the share button. So this is how you can save links for yourself and also how you can, send them to somebody else like, hey, I need help with this. Somebody have a look. Like maybe I would send a link being like, hey, can you look at that part that I got a little stuck at the end? I'm gonna share this. I'm gonna go over to our little proofreading sheet. We're pretending it's done, but I'm gonna select work in progress here because I'm still working on it. And I'm going to hit the link here so that I can come back. Need anything, you can put notes. We have help and comments. We also have ways that you can, say like, hey, I need help here. And you or fellow peers or as admins can come in and help out. So there's also some other things where like if you definitely hit Glia, you might get some of these segments maybe might be a little Glia, and it'll be pretty obvious because Glia looks pretty much the same as what you see in Outlier. So that's the incredible thing about it. Um, so it's really not terribly, these are kind of nice for starting out right now because they're, they can be longer tasks. You can come back to it. That's why I even put work in progress in the demo today. Um, but it's not like a whole cell you're taking ownership of that's going to take a while for you to proofread. These can be, shorter tasks and do reasonably in a set time, kind of as you're familiar with cubes, because we know that, sometimes it's nice to just sit down, do cubes, take a little breather from something, and then go back to whatever you were working on, and then maybe take another breather on iWire. And we know how there's different flows and preferences when we're kind of doing these reconstructing these neurons here. And it's, this is very helpful, even the little bit that I've done right here. So thanks for joining, Jihao. And that's one reason why I'm wrapping up real quick here before I fully finish proofreading here. But you can see that it's mostly for the actual actions you need to take to proofread that neuron. It's very nice and straightforward. You just merge and you cut, and that's kind of how you go. I will take a couple minutes of questions here, but I think it'll be a little more useful for when we can do a little bit more in depth, and maybe when folks can read through some of the guides. We have some up on the blog, especially with introductions to like, what are all these buttons? I know there's a lot going on here. As I mentioned earlier, this is like kind of the industry standard for connectomics for looking at these full data sets now, this particular software. So we're trying our best to make it as user-friendly as we can. So
Do they already have access to Peer right now?
I'm going to grant it to them after this call.
Want to do it like the Flywire ones? We could do it right now. Should we?
I can, well, that's one that I gotta go into a database and do
Okay,
it.
it's
So
a nice little GUI. There's like a with a
yeah, I don't. It's a little bit code-like. So yeah, but right after this, those of you who are already familiar with Flywire and the bank, we'll just grant you right-on access because it's pretty much similar. The nice thing is the bank is already in this version. We call it Spelunker of NeuroGlancer. So it's kind of like you can think of it as like, you have the operating system Windows, but then it has a number after it, right? We're on Windows 11. Well, this is the Spelunker version of NeuroGlancer. So, or I guess Mac, right? They call their versions like different names like Mojave and stuff. So, um,
All right. Well,
yeah, and if you, yeah, perfect. You place play.peer.ai, and that'll have your little user profile too once you get access in. So I'll stop sharing my screen. Um, So I know there are probably a lot of questions and a lot of things, and as we go through, I will be doing Zooms or Twitches or things like that, and we definitely will be interacting with you as best we can. We have a discussion board forum for where we can ask questions to as well, and kind of get people oriented and used to using this new thing and jumping into a whole new data set where it's kind of exciting to be able to, we're still working on mouse here, but we get to explore cells that are related to memory. And I wasn't even thinking about location.
yeah, it's really exciting. I mean, the hippocampus performs so many different functions. I mean, if you guys don't use ChatGPT, now's a really great time. Like even Jihao was he's an expert in this stuff, and like he's still using ChatGPT because it's difficult to summarize what the hippocampus does because it does so many things, because it's like involved in forming new memories, but also referencing what you already know with what's happening to you in the present and relating and modifying it, like and it also performs a lot of like pattern recognition, which is very like abstract, but it's pretty critical to like every type of thought, and basically interacting with the world. You're like predicting what's going to happen next, and that's basically done through pattern recognition. And we know like so little about like how all these systems work and how they interact like with the other parts of the brain, but it's really going to be like a groundbreaking leap, like once we get more regions of the hippocampus, like what the time frame that Jihao was mentioning, like 8 years from now. That's the first time I've heard that timeline too, so I'm really excited about it. But if we have a whole hippocampus in that period of time, like it will just reveal the pathways that are involved in sort of like the command center of presenting experience in the world, and translating that into like long-term connectivity in neurons, which I don't know. I think that's so cool. Like, I don't know. Definitely, neuroscience is the best field of biology.
I think we might be a little biased, Amy.
The most biased person in the world about the brain because I'm really obsessed with it. It was fun to make those slides in the beginning and look at how far things have come. Because I know, especially in the past few years, there hasn't been changes to iWire, and a lot of the changes that have been happening at lab have been through um, Neuroglancer, which is like that 3D viewing platform that allows you to like ingest and process data sets. And Chris has been on this call. He's like the main developer. You know him from iWire, but he's been working on Neuroglancer and building like this new branch of Spelunker that is that's peer. And once you get access and go to peer play.peer.ai, you'll see like your profile. But it's exciting to kind of open the next chapter because again, this is like the first of at least two data sets that are coming out this year. Like I think there's another data set that's coming in like 2 months, maybe like there's a lot happening at the lab. Finally, the pipeline is converging so that there's something pumping out of it. And so this is the beginning. So thank you for being at another beginning with us as we kind of venture into the next chapter of Connectomics. Any questions before we sign off? All right. Well, I'll let you guys go so you can be granted your access. Please send us suggestions and feedback. And I know Chris, Chish, oh my God, I learned how to say it. Hold on, I'm gonna say it right. Krzysztof, I think.
Oh, the Polish pronunciation.
Yes, Krzysztof has been working in Spelunker, so I'm sure he'll be in there, looking at and making new features, as will we. So yeah, all right. Well, nice to see you guys, and we'll post this video up. And then there's a detailed proofreading session next week. What is it, Wednesday at 11?
Yeah, and that'll also be recorded. And also, Celia, I'll have a link to, we'll post that up on, we have a peer YouTube channel, and Celia has another kind of overview of how these tasks all go that kind of is very similar to what I was just demoing briefly today. And she completes all the branches, so that's a good thing. I never know. That's the one thing. Sometimes you get a branch and you don't know where it's going or how long it's going to take. And you know what? That's the same with a cube, right? Sometimes you get a cube and you're like, oh, this cube turned out to be a big cube. And I think that's some of the joy, right? You never know quite what you're going to click and get and like extend out and grow in terms of these cells.
Just surprise.
Yeah.
Well, thank you guys. Thank you guys for joining, and we'll look out for an email. We'll be emailing you very soon, granting you access
Hmm.
here. Welcome.
Yep.
Bye.